All right, so our first speaker is Yang Dan. She had a PhD from Columbia, a postdoc, two of them, Rockefeller and Harvard. She's now professor and Howard Hughes investigator at Berkeley and a member of the National Academy of Sciences. She's interested in neural circuits underlying sleep and executive control. So we're going to hear more about that in a talk called Motor Theory of Sleep-Wake Control.
Is this turned on? Can you guys hear me at the back? Yeah? Okay, great. So first, I'd like to thank Nikos and the organizers for inviting me to this wonderful meeting. Now we've all had a wonderful lunch, feeling full and happy, so this is a good time to talk about sleep. Sleep is an essential innate behavior. We all know how to do it when we're born. No learning is required. As far as we know, all animals sleep, including flies and worms, and even jellyfish with a very primitive kind of nervous system. And sleep deprivation causes many problems, including emotional impairment, cognitive impairment, and other issues like impairment of the immune system and cardiovascular diseases. Mammals, birds, and probably all vertebrates have two distinct types of sleep, and they can be both distinguished from wakefulness based on EEG and EMG recordings. During wakefulness, the EEG is desynchronized, low amplitude, high frequency. EMG shows high muscle tone. During non-REM sleep, also called slow wave sleep, you see these large amplitude slow waves, reduced muscle tone. And during rapid eye movement or REM sleep, the EEG is actually similar to the wakeful EEG, but there's a complete skeletal muscle paralysis, which prevents us from acting out our dreams. So if you plot some simple parameters of the EEG and EMG, these three brain states fall into three distinct clusters, and that makes it pretty straightforward to quantify sleep. So the normal sequence is that we first go from wakefulness to non-REM sleep, and then we cycle between REM and non-REM sleep. So what controls sleep and wakefulness? Pioneered by Moruzzi and McGowan, who discovered the ascending arousal system, now we know that there are multiple neuromodulatory systems that promote wakefulness, including noradrenaline, dopamine, serotonin, and acetylcholine, and histamine. And also there are some neuropeptides, such as orexin. So we heard about these neuromodulators, right? So they're not just for wakefulness, they're involved in many other behaviors, but in terms of sleep-wake control, they all promote wakefulness. Now, in addition to these modulators, there are also many conventional glutamatergic and GABAergic neurons in the brainstem, midbrain, hypothalamus that promote wakefulness. In fact, it's really not that difficult to find neurons that promote wakefulness. If you think about it, a loud sound can wake us up, right? The kind of vibration stimulus that Ranolfo talked about this morning can probably wake us up. In contrast, it's much harder to find neurons that control sleep. So this was from a paper published 14 years ago, and they highlighted a single brain region called the preoptic area, the POA. So the idea is that the POA neurons promote sleep by inhibiting a lot of these known wake-promoting neurons. So my lab started working on sleep about 8 or 9 years ago, and so we're trying to figure out what controls sleep. We set out to two very simple goals. One is to find sleep neurons, and the other is to map their connections. And today I'm going to focus mostly on this first question, how to find sleep neurons. We have two basic criteria for sleep neurons. First, they need to be sleep-promoting, meaning that their activation should increase sleep, their inactivation should decrease sleep. Second, they should be sleep-active, so that they need to fire more during sleep in order to do their jobs. So we start in the POA. Von Economou, almost a century ago, studied post-mortem brains of human patients with sleep disorders. And he found that lesions in the preoptic area, this red circle here, are associated with prolonged insomnia. So that's what led to this idea that the POA is a sleep center. And then in the 1990s, Clifford Saper's lab and some other labs used CFOS immunohistochemistry. And so what they found is that a subset of the POA neurons are sleep active. And they're mostly GABAergic, and that's what led to this idea that the POA sleep neurons promote sleep by inhibiting the wake neurons. So using retrograde labeling and gene profiling, we have found a number of neuropeptide markers that are preferentially expressed in the sleep neurons. And taking advantage of mouse genetics, we were able to activate those neurons specifically and show that their activity can promote sleep. But that wasn't the end of the story. It turns out that there are other sleep-promoting neurons outside of the POA. So we first became aware of this from the work of Franz Weber, a brilliant former postdoc in my lab who's actually German. And later, Franz was joined by Johnny Doe, a graduate student. So Franz came to my lab really searching for the REM sleep neurons, the neurons that promote REM sleep. So he indeed found a population of GABAergic neurons in the medulla that very powerfully promote REM sleep. But when Franz was trying to find the postsynaptic targets of these REM neurons, he found these neurons in this region called the VL pack. Ventrolateral periaqueductal gray is a handful, so we just call it VL pack. So here, Franz expressed channelodopsin in the GABAergic neurons in the GAD2-Cre mice. And here is our standard experimental protocol. So this is the EEG power spectrogram. So if you see this sort of broadband low-frequency activity, the animal is likely to be in non-REM sleep. And if you see this thin gamma band activity, 8 Hz, the animal is either awake if you see high EMG activity, or in REM sleep if the EMG is flat. So this blue shading indicates a laser stimulation period. So in this case, each trial is 5 minutes, and there's a random inter-trial interval. So this is the laser-triggered average of the 3 brain states that we averaged across all the mice that we tested. So you can see that when we turn on the laser at time 0, there's an immediate and strong increase in non-REM sleep, this gray trace, decrease in both wakefulness and REM sleep. So this effect is actually very reproducible. So here I'm showing the color-coded probability of each of the 3 brain states before, during, and after laser stimulation for the 12 mice that are tested, some by Franz and some by Johnny. And you can see that in each single mouse during the laser period, REM and wake probability is reduced, and there's a clear increase in non-REM sleep. So it seems like what's going on here is that these yellow or orange neurons are non-REM promoting neurons, so they're important for going from wakefulness to non-REM sleep. And the blue REM neurons in the medulla that Franz discovered push the animal from non-REM sleep to REM sleep by inhibiting these non-REM neurons. So the problem here is that, like I said, for a long time we thought that the POA is the sleep center, right? And yet now we found these VL pack neurons, which are quite far from the POA, and they very strongly promote non-REM sleep. And at about the same time, there were a couple of other groups who also reported some non-REM promoting neurons outside of the POA. But they seem to be sort of popping up at random locations here and there, and we don't know how many more are out there and whether there's any logic to this. So a few years ago, we decided to start a whole brain screening for sleep neurons. So like I said, we have 2 criteria for sleep neurons, and they correspond to 2 strategies for screening for sleep neurons. We can either screen for sleep-active neurons and see if they're also sleep-promoting, or we can screen for sleep-promoting neurons and see if they're also sleep-active. So I'm going to spend the rest of the time showing you some examples of each strategy. So first, we screened for sleep-active neurons using a strategy developed by Li-Chun Luo's lab at Stanford. So we crossed two mouse lines together. One expresses CRE-ER under the FOS promoter. So FOS is an activity-dependent early gene. So the active neurons should express CRE-ER. The second mouse line expresses GIP in a CRE-dependent manner. We divided these mice into two groups. One we call the SD group, so we sleep-deprive them. The idea is that the wake-active neurons that are active during the sleep deprivation should express CRE-ER, and the injection of tamoxifen should cause the CRE-ER to enter the nucleus, and that causes the permanent labeling of the wake-active neurons with GFP. In the second group, we first sleep-deprive them for 6 hours to make them really sleepy. And then if we leave them alone, they go into recovery sleep. So they sleep very well during this period. And the injection of tamoxifen allows the labeling of the sleep-active neurons. So when we compare these two groups of mice across all the brain regions, we found that some regions express more GAP in the RS group, some express more in the SD group. But our top candidate, this region, with the highest relative expression of GAP in the recovery sleep group is right here. So this is the sleep-deprived group, and this is the recovery sleep group. So you can see a lot more GAP here. So when we found this region here, we have to say that we're very, very excited. And that's because in the sleep field, everybody knows about von Economo, who proposed the POA as a sleep center. But in fact, several decades before von Economo, Mausner, this is the guy who discovered the Mausner cells in the fish, so he actually proposed a sleep-regulating center near the ocular motor nucleus. So the third nucleus is like right here and here, right? And so this region that we found is pretty much what Mausner proposed. Tragically, Mausner died just a few years after he proposed this. Now, after he died, the idea of a sleep center remained very influential. In fact, that's what motivated von Economo to search for his own sleep center. But the particular center that Mausner proposed was largely forgotten. So when we found this, I actually didn't know about this paper. It's actually my postdoc, Jill, I showed her picture in the last slide. So she dug up this paper, and I realized that, hey, there's actually a history here. But obviously, even though we see a lot more GFP neurons, it's not all the neurons in that region. So we have to figure out who these GFP neurons are. So for that, we did gene profiling. Now, this reporter mouse line that we used expressed not only GFP, but also this large ribosomal protein that normally binds to mRNA to do translation. So if we use an antibody against GFP, to pull down this ribosomal protein, we can also pull down the mRNA attached to them. And so then we can do sequencing. So this is what the data look like. This horizontal axis is the relative expression level of the gene, right? So each dot is a gene in the putative sleep-active neurons relative to this entire chunk of tissue that we cut out. And the vertical axis is the p-value. So if we zoom into this box, right, which are the genes that are highly significantly enriched, there are a whole bunch of genes. One of them is the one that we overexpress, right? So this is a positive control. There are many other genes. I have no idea what they do. But we decided to focus on neuropeptides because neuropeptides are functionally important, and also just because there happen to be a lot of CRE lines for neuropeptides. So there are three neuropeptides, CALCA, CALCB, and galanin. And when we looked at their expression, it turns out CALCA and CALCB overlap by more than 80%. So they're pretty much labeling the same neurons. Galanin labels a very small number of neurons, and more than half of them also express CALCA. So we decided to focus on CALCA as a peptide. And indeed, when we compare CALCA expression with the GAP labeling of the putative sleep-active neurons, there's a high percentage of overlap. So we decided to look into the CALCA population. So first, we wanted to know whether these neurons are indeed sleep active. So even though CFOS is an activity-dependent gene, it's not a perfect reflection of activity. So we wanted to record directly from these cells. So for that, we expressed channelodopsin specifically in the CALCA neurons using a CALCA Cree mouse generated by Richard Palmiter's lab. And for recording, we use an Optrode with an optic fiber surrounded by several bundles of wires for recording. So here is one unit. You can see that it's a very nice single unit. Every time we give a brief laser pulse, this little blue dot here, 5 milliseconds, it evokes a spike very reliably at short latency. So we're pretty confident that this is a neuron that expresses channelodopsin, therefore it should be a CALCA neuron. So once we find a neuron like this, which is actually quite difficult, if you find one cell in a week, it basically makes your week, right? So it's a very hard experiment. But once we find it, we just turn off the laser and record a spontaneous activity. So here's one cell, and you can see that the firing rate is high during non-REM and low during both wake and REM. And here's a summary of 30-something cells. So each line is one cell across the 3 brain states, the firing rate. And we also summarize their activity in this 2-dimensional plot. This axis is the firing rate difference between non-REM sleep and wakefulness, and this is the difference between REM and wake. So most of the identified units, the blue units, are on the right-hand side, indicating that these neurons are more active during non-REM sleep than wakefulness, even though the relative activity between REM and wake is quite scattered. But if you compare with the unidentified units, these gray dots here, so these are recorded in the same location, but they don't respond to the laser stimulation. We don't know who they are, but you can see that many of them are on the left-hand side, so these are the wake-active neurons. So it seems like most of the CALCA neurons are non-REM active, so this is our first criterion. Now, are they non-REM promoting? So here we use channelodopsin to activate these cells, right? So this is our standard laser triggered average, and you can see there's increase in non-REM sleep, decrease in both wake and REM. And if we inactivate these cells using a light-activated chloride channel, IC++, we saw a decrease in non-REM sleep. So this is just a quick optogenetic activation, right? So each trial, in this case, is only 2 minutes. But the argument is that sleep is a slow process, right? We don't just sleep for 2 minutes per trial. So we also did a slower manipulation, which is using the DREAD technique. So in this case, when we express the excitatory DREAD into these CALCA cells and inject CNO, which activate this GPCR, then during the first 2 hours, we see an increase in non-REM sleep compared to the control. And if we inactivate these cells using the inhibitory DREAD, we saw a decrease in non-REM sleep. So basically, both the activation and inactivation, optogenetic and chemogenetic techniques, everything checks out. So these neurons are non-REM promoting. So this is our second criterion. Okay, so we use CALCA, which is a neuropeptide, as a marker, right? But it turns out that the CALCA neurons, that population contains 2 subpopulations. About 60% of these cells are glutamatergic, and the other 40% are actually cholinergic. So I'm not going to show you the data, but just tell you that it's actually the glutamatergic population that promotes sleep. The cholinergic neurons, when you activate them, they cause pupil constriction, which, by the way, happens during sleep, right? But activating them alone does not promote sleep in any strong way. So it's the glutamatergic neurons that are doing the job. But it turns out that the CALCA neurons are not the only glutamatergic neurons that promote sleep. There's another population. So these cells express the peptide CCK. The CCK neurons are almost 100% glutamatergic, and they're entirely separate from the CALCA cells, right? So the CALCA cells are these red cells, and the green cells are the CCK cells. Now, the CCK neurons, if you activate them, you also see increase in sleep, inactivation decrease. So these neurons are also sleep promoting. So now we have two populations, right? They're sitting right next door to each other. The question is, do they talk to each other, or they're just, barely just neighbors? So for that, we did brain slice recording. In this case, we expressed channelodopsin in the CCK neurons using CCK Cree mice. And then to identify the postsynaptic cells, we basically suck out the cytoplasm from the recorded cell and then did a single cell RT-PCR. So this happens to be a CALCA neuron, and we see that activating the CCK neurons causes this very strong excitatory input, which can be blocked by the AMPA receptor blocker. Conversely, if we activate the CALCA neurons, we saw this very strong excitation in the CCK neurons. So basically, these two neurons do talk to each other. So here's a quick summary of what I showed you so far, right? We found two populations of glutamatergic neurons in that region, the CALCA and CCK neurons, which actually are reciprocally connected. I'm not going to show you the data, but just tell you that both populations project to the POA, right? So this is the well-known sleep center. And one of them also project to this region in the medulla called the GIV. So if you activate each of these axon projections, we also saw increase in non-REM sleep. And by the way, both of these populations of neurons happen to inhibit a whole bunch of wake-promoting neuronal populations. So that suggests that actually the ventral portion of the PAG, these neurons, they form sort of an excitatory center for sleep promotion. They're just upstream of these GABAergic sleep-promoting neurons. So this is the first technique, right, screening for sleep-active neurons. Now I'm going to show you some examples of screening for sleep-promoting neurons. So we have this very simple-minded idea. We're going to start with the wake-promoting neurons. Like I said, we know a lot of them. And we're going to do retrograde tracing to find inhibitory neurons that are upstream of these wake neurons. So in this case, we're looking for GABAergic neurons. Because if you're a neuron that inhibit the wake-promoting neurons, then chances are you could be promoting sleep yourself. And once we find these GABAergic sleep neurons, we're going to trace one more step back and look for their excitatory input. Because if you excite other sleep neurons, then maybe you promote sleep yourself. So this was done by Chen-Yan Ma, a very talented postdoc in the lab. So Chen-Yan targeted a whole bunch of known wake-promoting neurons. And so she was looking for GABAergic regions that not only project to one wake-promoting population, but to a whole bunch of them. So she wants to look for neurons that broadly inhibit many wake-promoting neurons. So Chen-Yan targeted 7 populations of wake neurons, including histaminergic, cholinergic, noradrenergic, glutamatergic, and GABAergic in 7 different brain regions. And these are several brain regions with relatively broad GABAergic innervation of these wake-promoting neurons. And that includes the POA, right, which is consistent with the textbook version. But to our surprise, our top candidate that came out of this screening is actually the central nucleus of the amygdala. So here's just an example of each of the 7 populations she targeted. And if you look at this little square here in the CEA and you blow it up, you see a whole bunch of rabies virus labeled neurons that also express the GABAergic marker GAD1 or 2. So again, we already know from the people who study fear learning that the CEA contains many different cell types. So we have to figure out these putative, sleep-promoting neurons. Who are they? So for that, we again did gene profiling using pretty much the same technique. So again, here's the data. And if we zoom into this little box here, we see a bunch of genes, including 3 neuropeptides. And when we looked at expression of these peptides, NTS, neurotensin, and tachykinin 2 overlap by more than 80%. So they're pretty much labeling the same population. Neurotensin and prodynorphin also overlap by about 50%. But because neurotensin has a more localized expression in the CEA and it doesn't label the surrounding regions, we decided to focus on NTS as a marker. And indeed, if we look at the NTS expression with the rabies, the retrogradely labeled cells, we see a very high percentage of overlap. So here we tested the effect of activation and inactivation of the NTS neurons. There happens to be another NTS Cree mouse. So activation, increase in non-REM sleep, inactivation, decrease. So again, the NTS neurons seem to be sleep promoting. We also did Optrode recording, right, the same technique. So here's one example cell, and here's a summary. The blue dots, the identified NTS neurons, they're mostly in the upper right quadrant, indicating that they're more active during both REM and non-REM sleep compared to wakefulness. So they're sleep active. This is our other criterion. Okay, so now we found one population of GABAergic sleep-promoting neurons. And now we're going to trace one step back and look for their excitatory input, glutamatergic input. So when Chen-Yin targeted the CEA NTS neurons, she found several brain regions with strong glutamatergic input to them. One region shown here, right? So you see these rabies-labeled GAP neurons that express VGLU2, which is a glutamatergic marker. So who are these cells? Okay, so first of all, what is this region? So this is an Allen mouse brain atlas. This red region here, expressing VGLU2, is this red region here. So this is the posterior part of the thalamus. And unfortunately, VGLU2 is not a good marker, right? Because you see that it's expressed in the entire thalamus. But these rabies-labeled cells are only found in this vertical strip right at the border of the thalamus. So we have to figure out who they are. Now, in this case, we just made a lucky guess, right? So we thought, okay, so NTS we found is a good marker for the CEA. What about NTS, maybe in this region? And it turns out if we look at the NTS expression, so this is actually from the Allen database, neurotensin is exactly expressed in this vertical strip. And when we compared it with the rabies-labeled cells, there's a high percentage of overlap, right? So it seems like NTS happens to label that population as well. And when we activated NTS cells in this region, again, activation, increase in sleep, and inactivation, decrease in sleep. So these cells also seem to be sleep promoting. So we said, okay, now we have two populations, right? They're connected. And the first is GABAergic, and the second is glutamatergic. And yet they both express NTS. So before that, we were just using NTS as a marker. We didn't care what NTS was doing, right? But now the question is NTS peptide itself doing something, right? So I was asked that question many times, so finally we decided to do it. So Chen-Yin, who's really, in my mind, a genius in molecular biology, she designed this very complicated construct, and I can't really explain to you in a clear way, right? But basically what it does is that it expresses a guide RNA for neurotensin, and it also expresses channelodopsin and M-cherry. So if we inject this into the NTS cells, inject into the CEA of the NTS Cree mice, what this guide RNA does is that it allows this CRISPR technique to basically knock down NTS, right? So this guide RNA guides Cas9 kinase so that it sort of nips the DNA so that there's an error in the NTS gene. Therefore, NTS expression is severely affected. So this is the control, and this is the NTS knockdown. So we did that for both the CEA and the posterior thalamus. And what we found is that, so what this is showing is that it's the effect of change in sleep caused by optogenetic activation of the NTS neurons. So this is the control, right? So like I showed you before, if you activate the CEA NTS neurons without knocking down NTS, you see this big increase in non-REM sleep. But in the NTS knockdown animals, you see that the effect of channelodopsin is greatly reduced. The other experiment we did was to knock down, instead of NTS, we knocked down GABAergic synaptic transmission. So we knocked down this gene, which is VGAT, the vesicular GABAergic transporter to load GABA into the vesicles. When we did that, there's also a very strong decrease in the sleep-promoting effect. Now, for the posterior thalamus, knockdown of NTS basically eliminated the sleep effect, and knocking down VGLUT2, which affects glutamatergic transmission, also largely eliminated the sleep effect. So the conclusion is that basically both the neuropeptide and the traditional transmitter, either as GABA or glutamate, are important for the sleep-promoting effect. So here we found two populations of NTS neurons, and we actually got quite interested in NTS because it turns out that the NTS peptide, which was discovered in the 1970s, does all those wonderful things that happen during sleep. It slows down the heart, it drops the body temperature, reduces the blood pressure. All of them happen during non-REM sleep. So we thought that this is too much of a coincidence, right? So maybe there are other NTS neurons that are important as well. So Peng, another postdoc in the lab, found this other population by basically going through the Allen Institute database. So you see this NTS neuron, the cluster, again back to the pack, right, which I talked about earlier. So if you activate these pack NTS neurons, which happen to be glutamatergic again, huge increase in non-REM sleep, inactivation, decrease in sleep. And using the tools that Chen Yan developed, I forgot to mention, so Peng also did Optrode recording, but just here for change, I'm going to show you some calcium imaging data. So we imaged the VL-PAC NTS neurons through this microendoscope with the miniaturized microscope, and you can see that the calcium activity is also locked to brain state, right? So high activity during non-REM and decrease in both REM and wake. And using the tools that Chen Yan developed, Peng also did the NTS and VGLUT2 knockdown, and in both cases, compared to control, there's a much weaker sleep-promoting effect. So again, both glutamatergic transmission and NTS are important for sleep. Okay, so a quick summary of what I showed you so far, right? So we screened for sleep-promoting neurons, and we found NTS neurons in the CEA, GABAergic, posterior thalamus, glutamatergic, and VL-PAC, also glutamatergic. All of them promote non-REM sleep, and both the peptide and the traditional transmitters are important for this function. And there are two other things that I kind of wanted to highlight. One of them is that NTS turns out to be a very general marker for sleep-promoting neurons. I told you about these three regions. There's actually another region in the subthalamic nucleus. I haven't shown you in the data, but I'll show you later. So all of these, we have shown that they promote sleep. The other thing is that the ventral pack really seems to be an upstream non-REM promoting center because we have found three populations of glutamatergic neurons expressing different peptides, and all of them promote sleep, and they also project to these known GABAergic sleep promoting centers. So that seemed to be an important upstream sleep promoting region. Okay, so back to Chen Yan's screening, right? So I told you about these first two regions, and what about this third region? So I'm going to quickly take you through this third study. So the third region that came out is the substantia nigra pars reticulata. So this study was done by Dan-Chen Liu in the lab. So as you heard from Anne's wonderful talk yesterday, I think, right? So the SNR is actually an important part of the basal ganglia circuit controlling movement. And yet it came out of this sort of screening for sleep neurons. So in order to understand how the neurons could be involved in movement control and sleep, we decided to first take a look at the motor behavior of the mice in their home cage. So here's a mouse in the home cage. In addition to EEG and EMG, we also made a video recording. And our collaborators in the Institute of Automation in Beijing, they used deep learning, which is all the rage now, and we also wanted to, jump into the action. So they trained this convolutional neural network called UNet to segment the image. So this is the raw image, and this is where the network thinks the mouse is, and you can see it's pretty good. So once we do this, we can easily extract some simple parameters. One of them is translation. So we look at the center of mass of the mouse in two different time frames, and this distance, we call it translational movement. The other is the total number of pixels that are different between the two time frames occupied by the mice, right? So in this case, it's the total number of blue pixels. The mouse was like facing this way and then that way. So if we plot these two parameters, they seem to fall into 3 clusters. The dark green cluster corresponds to locomotion. So the mouse is like running around, right? There's a lot of movement. The light green corresponds to non-locomotor movement, right? So it's either eating, grooming, or some posture adjustment. The third cluster is basically immobility. But when we look at the EEG and EMG recordings correspond to this third cluster, they're actually 2 different states. One is quiet wakefulness, and the other is actual sleep, right? So just the fact that they're not moving doesn't mean that they're necessarily asleep, because you can tell from EEG and EMG signals. So this is just a video. I think I'm running out of time, so I'm not going to show you the video. Oh, actually, it runs anyway. But just to point out that when we look at these 4 behavioral states, you can see that as the animal goes from locomotion to movement to quiet wake to sleep, the delta power, the low-frequency activity in the EEG, increases progressively. So the brain is getting less and less aroused. And the EMG activity decreases monotonically. Now, when we looked at the transitions between these different states, they're not random at all. So if the mouse was in the locomotor state in the previous moment, they always transition into non-locomotor movement. We have never seen a case where the mouse was like running in one moment, next moment, bam, they fall asleep. It just doesn't happen, right? So I mean, in narcoleptic animals, you might see this, but in normal mice, you don't see that. And if you start with non-locomotor movement, either they go back to running or they go into quiet wakefulness. They also don't fall asleep right away. So these are the transitions. So it seems like you can almost rank these 4 states in a single chain, right? And most of the transitions are between neighboring states. So this is the transition matrix, and all the non-zero transitions are either right above the diagonal or right below that, right, between neighboring states. The only exception is that sometimes they go from sleep right into movement by skipping the quiet wakeful state. Okay, so now we have this behavioral characterization, and now we want to move into neurophysiology. So it turns out that in the SNR, there are actually 2 types of GABAergic neurons. Now, if we use a PV Cree mouse and injecting the Cree-dependent channelrhodopsin virus, we mostly label these lateral cells that are PV positive. But if we use a GAT2 Cree mouse, we mostly label the medial SNR neurons that are PV negative. And when we did recording from the PV positive cells, again, Optrode recording, we see that the PV cells are actually what we call movement activated. So you see that they're most active, so each line is 1 PV cell. They're most active during locomotion, less activity during movement, gradually down, least active during sleep. But when we looked at the GAT2 neurons, they have this opposite behavior, right? So they're least active during locomotion, most active during sleep. And this is just the average of the PV population and the GAT2 population. You can see that their trends are pretty much the opposite. Now, what about activating these cells? So if we activate the GAT2 neurons, we saw a huge decrease in locomotion and also movement, and pretty clear increase in sleep. But activating the PV cells, we saw a very minor effect. And this is just a summary of the GAT2 versus the PV effect. And when we inactivated the GAT2 cells, huge increase in movement, huge decrease in sleep. And again, the PV effect is much, much weaker. So what we think is going on, so it seems like the PV neurons are movement active, and their activation mainly terminate movement, but they have no effect on sleep. But the GAT2 neurons are movement suppressed, and their activation suppress movement and enhance sleep. So what we think is going on is that the PV cells are probably important for action selection, right? So for example, right now, my desired action is to give the talk, so I have to suppress my need to run out and get a coffee, right? So if you want to perform one action, you got to suppress all these other distracting actions. And PV cells might be doing that, right? So they're active during movement, but their activation suppress movement. The GAT2 neurons seem to be controlling the overall state of movement, overall how much movement you're making, right? And that's why the more active they are, the less movement we have. Okay, so we took a closer look at the effect of activating the GAT2 cells. So it turns out that even though we saw a huge decrease in movement and increase in sleep, we're actually not directly, making the animal go directly from movement to sleep. Instead, what we see is the increase in these transitions, right? So these red squares above the diagonal and decrease in the other transitions. So basically what we're doing is to bias the probability of the natural transitions down the chain, right? And inactivation pretty much has the opposite effect. So we're not creating artificial transitions. We're just biasing the transitions, either go one direction or the other. So I think that this provides some insights into how the movement control neurons could also be involved in sleep, because you're kind of, biasing the behavioral transitions. Okay, so we also looked at the projection targets. Now, the PV cells project to the motor part of the thalamus, the superior colliculus, this locomotor region. And in addition to these movement regions, the GAT2 neurons also project to some neuromodulatory regions, including the dorsal raphe and the locus coeruleus, which the PV cells don't project to. And so we also looked at the collaterals, and it turns out that it's not the different subpopulations of GAT2 neurons that project to these different regions. Actually, the same neurons can send different axon collaterals to the motor regions and also to the neuromodulatory regions. And I'm just going to skip the data here. Okay, so very quickly, so we also searched for their upstream targets, and we found these STN cells, which happen to be NTS positive, and then activating these cells also increases sleep. I'll be done very quickly. Okay, so this is a summary, right? I pretty much told you I'm not going to read it through. These are all the data, and I'm just going to spend 1 minute speculating. So when we started working on sleep 8 or 9 years ago, the first few years were very frustrating. We were not finding any sleep neurons. And now with this kind of screening, we're finding a lot of sleep neurons, as I was telling you today. Now the question is there any logic to this, right? Or is it just a random collection of cells? Now, we actually think that there's a very simple logic, and we call it the motor theory of sleep. So if someone is asleep, the first thing you can tell is that that person is not moving. And in fact, for all the non-mammalian animal models, sleep is scored entirely based on the lack of movement. So this kind of visible movement, right, body and limb, is controlled by the somatic motor system. Now, there's another type of movement, harder to see but equally important, including heartbeat, breathing, and things like that. They're controlled by the autonomic motor system. So when we fall asleep, the somatic motor system is pretty much shut down. The autonomic motor system also slows down a lot. Now, with all this screening, even though we were looking for neurons that are regulating brain state, all the neurons that we found so far seem to be associated with these two motor systems. For example, the STN, SNR pathway, right? So this is part of the basal ganglia for somatic motor system. The PAC, which I made a big deal about, is an important part of the central autonomic network. The POA, part of the hypothalamus. The amygdala I told you about, and even the nucleus of the solitary tract, which we're characterizing right now. So it seems that instead of being separate, the sleep control system deeply infiltrates these two motor control networks. On the other hand, the sleep neurons seem to be separate from the neurons controlling specific movement, like in the SNR, the PV cells regulate movement, but the GAT2 neurons actually control motor states. The same state neurons also send axon collaterals to these brain regions regulating brain states so that they can coordinate the arousal level and the amount of movement. So this view of how sleep is controlled also has an implication for the question of why do we sleep, right, which I haven't talked about so far. So we tend to think that sleep is about the brain, right? We shut off sensory processing, we lose awareness of the environment. But it seems like this kind of circuit, equally importantly, is about suppressing movement. So that's probably the function is to facilitate processes that are incompatible with a high motor state. It probably includes synaptic homeostasis, which we'll hear about in the next talk. But I think it's also about cellular repair. Now, just to be clear, I don't just mean repair of muscle damages caused by intense exercise, which, by the way, is actually very important for athletes. I think that neurons in the brain are also damaged somewhat mildly during prolonged arousal, and those cells need to be repaired during sleep. So that's it, and here are the people who actually did the work. I think I mentioned their names. We also had many collaborators who helped us with various techniques, and thank you for your attention.
Thank you. We have time for one question while Giulio sets up. The very back there. Someone have a microphone for him?
May I? Oh.
Very interesting. Is it good? Very interesting. Thank you. When I learned about sleep, the consensus was more or less the brain, cortical slabs, even the isolated brain, is sleeping when it's not being awoken by wake centers. So the brainstem wake centers are required for wakefulness, and the brain sleeps as a normal state. So where do your sleep areas come in now?
Right, so for a long time, we actually thought that sleep is a passive process, right? So if you just eliminate the things that keep you awake, right, sensory stimuli, then you can fall asleep. But that idea pretty much has been discredited. And, you know, a very important event is von Economou, right? He showed that if you lose some brain regions, you actually can't fall asleep. You can eliminate all the sensory stimulation, but you still can't sleep, right? So that's actually the idea of active sleep promoting center.
Thank you.